Good morning. Great to have you all in the room. Thank you all very much for joining and taking the time to listen in today. And thank you very much to the organisers at Wholesale Investor for putting on the event. I'm delighted to talk to you today about Arovella Therapeutics. We are a biotechnology company focused on cell therapy, as we just heard from CAR-iNKT Therapeutics, which is a nice introduction. We are listed on the Australian Stock Exchange under the ticker ALA, and I'm certainly looking forward to taking you through our invariant natural killer T-cell platform. I will be making forward-looking statements, so please take note of our disclaimer. Now, Jen introduced nicely, and I can see there's common overlap in some of the imagery here, where why we're so excited about cell therapies in terms of cancer treatment is because of what she just mentioned, that patients that have taken these therapies experimentally 10 years ago are still cancer-free, which is why we're using the word cure and cancer in the same sentence, which is a lovely place to be. Now, over that time period, what we've also seen since approval of the two leading therapies, Yescarta and Kymriah, one's done over $1 billion in sales, the other over 500 million, and another therapy approved in 2021, in 2022 did sales of just under 500 million. So it is set to be a very important pillar of oncology treatment, and of course, a very big market, as Jen alluded to earlier. However, we do see that there is a really important need for a different approach to these types of therapies, because the current therapies, T cells, they must come from the patient. Okay, so we take the starting material from the patient, genetically modify that material so it then ends up being infused back into that individual. So that costs about $500,000 US per dose, which is quite cost prohibitive. Logistically, it's very challenging, and so it sets the scene to have a very high bar. So what we're actually working with, it's an invariant natural killer T cell. We will be able to take the therapy from what we call a healthy donor. We make the therapy and then we can freeze the therapy, and I'll talk more about that in a moment. Another challenge when you're making the therapy is what we would say just in time, is that you have a number of weeks before the patients can get access to that therapy, which means that some that have aggressive disease may succumb to the cancer before the therapy can be produced before administration. So our iNKT cells, we do believe are set to solve some of those logistical challenges, but we also think for the data that we've generated to date may have advantages in terms of efficacy. So this is how we would start. Instead of having the therapy produced from a patient, we start with that healthy donor. The way these therapies work is we actually isolate human immune cells. We then genetically reprogram them to produce something that's called a chimeric antigen receptor, or CAR. But I'd like you to think about it as a guided heat-seeking missile that will find, recognize, and trigger destruction of cancer cells. They get grown up into the billions, and in our case, vialed and stored in the freezer, where they can be pulled out and immediately used for administration. So the advantages of this are that we have healthier starting material, because these cells will not have seen cancer. They won't have seen chemotherapy or radiotherapy, so we think they might be more efficacious. We also know that it will be scalable. We'll have hopefully hundreds, if not thousands, of doses per batch, not N equals 1, which is very, very cost prohibitive. We know that we will be able to get patients access to the therapies faster because we don't have that manufacturing step. And it also removes the risk of manufacturing run failure. Once it's in the freezer, we know that that therapy is ready, available for use. So in terms of our particular targets, we've got three major pipeline candidates at the moment. ALA-101, we're developing for what we call CD19 expressing lymphomas. And so this is a common malignancy that's now across a range of blood cancers, and that's our lead program, and it's our most advanced program to date, which we're hoping to advance quite soon into the clinic. So we also are combining that therapy with another well-known biotechnology company called Emergence in Australia. They have an oncolytic virus platform, and we combine the two therapies together. And what we hope that will enable us to do is actually target a range of different solid tumors, which opens up an enormous market, given that solid tumors do account for about 90% of cancer diagnoses. And on the very right-hand side, we have our third program. It's called ALA-104. It targets something else that's found on the surface of cancer cells through what we call, as I said, mentioned earlier, a different CAR. We're expecting that will have promising activity against blood cancers like multiple myeloma and also a number of solid tumors. So an important feature of our platform is that we expect that we can continue to expand and evolve the pipeline by switching out those guided missiles and adding new CARs to target a range of different cancers, including both blood cancers and solid tumors. So in terms of the pipeline, we do have quite a busy, we've had a busy start to the year, and we'll have a busy 2023 ahead of us. For our lead program, as I mentioned, it's called ALA-101, which is targeting CD19 producing blood cancers. We are currently in the stages of optimizing and scaling up the manufacturing. Once that's been completed, we can submit the necessary documentation to regulators to commence phase 1 clinical trials and get these therapies into the hands of patients. For ALA-101 plus the oncolytic virus in partnership with Emergence, we're currently working through some proof of concept studies. We saw promising in vitro work already, so we're moving to what we call in vivo or animal models to take that a step further. And for ALA-104, we're currently working through target validation and also working to combine that with our iNKT cell platform to demonstrate proof of concept. So in terms of a biotechnology company, what's really important equally behind the technologies is the team. So we've worked very hard over the last 18 months to assemble a really purpose-driven team. So we have a phenomenal amount of experience across cell therapies. I'm not sure why Minnie's there twice, but she certainly does seems like she works 200%. But certainly a wonderful team dedicated to drug development, in particular cell therapies. We do have representation across Australia, but also the US, because we know that that's where a lot of the talent lies for these particular types of therapies. And only last week, we're delighted that we've appointed Dr. Tom Duthie as our new chair of the board. He's got a wonderful amount of experience, and he was formerly the head of investor relations and corporate development at a company called Certex, which was acquired for $1.9 billion in 2018. So delighted to have him join as our chair. So in terms of the company, we're currently capitalized at about 25 million, but over the course of this year, we completed a placement early in the year at 2 cents, and we're now back at 3.5. So we do expect that we've got good results coming up throughout the course of the year. So we're looking for some value accretion from that perspective. And we do have some excellent shareholders on the register. Merchant Group is our largest shareholder, so they've had a lot of experience across the biotech sector. And we do also have a number of good, supportive, high net worths and small institutional investors on the register as well. But certainly looking to welcome additional investors to the Arovella register that also understand the potential rewards that can come with an investment into biotech as well as the risks. And with that, I'd just like to say thank you very much for your attention.